Good afternoon, everyone. My name is Rich Win, and today I'd like to share with you an accurate cell detection method with minimum training effort. In a typical year, about 5 to 20% of the population will go under the flu, while most of us experience the flu and get over it quickly. Some people may experience further complications in which they have to go to the health center to get themselves checked up. And if the flu persists over time, they're going to need a blood test to see their health conditions. And in a drop of blood, the number of white cells can determine your health conditions. If the number between 5 and 25,000, you consider it healthy. If the number fall below 5,000, you're under a flu or some poisoning. If your body fight back, the number of white cells will significantly increase, which means you under a lot of stress, has viral infections, and drug intakes. However, if the number go beyond 50,000, then you may have leukemia or blood cancers. In 2009, there are 45,000 people in the United States have been diagnosed with leukemia. Even though treatments are possible, they had no known way to prevent leukemia. So natural killer T cell is a special type of blood cells that have been great interested for a researcher at the moment, because they think that these cells would move toward the cancer tumors and kill it. The biological question to ask is what would aid them to move toward the cancer tumors. In order to answer such biological questions and need cell analysis, and traditionally this process would be done by using manual detections of cell location in the image, and usually it will take over 1,000 mouse clicks to perform a single experiment. This process is tedious and expensive. So recently they have automatic cell detection methods that automatically determine these locations, which reduce the training effort the user had to put in. Instead of detecting cell, become to training the system to able to do it for them. So the number of effort had been reduced to 100 mouse click. But today I'd like to share you a method that only takes 10 clicks to achieve the similar performance at other methods. So we go from 1,000 clicks to 100 clicks, and now only 10 clicks. So let's look at a cell image in the microscope. These are red blood cells. They go under the vesicle. An automatic detection method with 100 clicks would be able to detect these cells pretty well. As you can see, the greens are correctly detected cells, the reds are incorrectly detected cells, and the yellow is undetected cells. If you reduce the number of training samples by 10% or 10 clicks, you would have a not reasonable accurate cell detections. And just imagine the consequence of having this incorrect analysis in your white blood count. That would not be feasible. However, we like to keep the minimum training effort, like 10 click, and with our method, the cell performance is better. So our method is an accurate cell detection method with 10% training effort, and we call it GATLab, or the Globally Active Transfer Learning for Adaptive Boosting. Let's look at the challenges that we had to face while we detected cells automatically. First, we had to deal with many cell types. Biologists studies not only one type but many types. Additionally, cells can look very different under a different capturing equipment or imaging protocol. The second is the problem with first-timer. Automatic detection method, when it look at new cell type, it is not be able to leverage on what is already known. It tackle the problem like a first timer, and the user had to train it from scratch. And the third challenge is the labor effort. How much work the biologist put in to train the system to get good detection results. We want to minimize the labor effort. So our previous research have been addressing the first and second challenge successfully. For the first challenge, we construct a cell size distributions that are quickly adapted to a new cell type using a statistical model about their appearance. For the second challenge, we able to learn from previous type now so that the algorithm can leverage on its existing knowledge and we don't have to train it from scratch. However, the third challenge have not been addressed until now. So let's look at the training process in more details. So we start with the user looking at the training image, and he or she can teach the algorithm by selecting cell and non-cell location at training sample for GATLab. To address the first and second challenge, we already have size distribution and knowledge from previous cell type built in of GATLab. But the third challenge about label effort still need to be investigated. And we look at the whole training process as it is, and we realize that the training process has been done almost randomly. The user just randomly keeps selecting the cell sample without knowing how helpful that training sample is, or maybe not as helpful. So we think we can improve this using an interactive approach, whereas the algorithm gonna return some kind of feedback to the user, so the user you can realize where the algorithm needs to learn next. And doing that, we help the user to reduce their training effort by only focus on selecting the most important sample, the most hard sample for the algorithm to learn. The hard sample could be a very faint cell that is difficult to see even by human eyes, or the noise particle that in the image that look like cells. These samples need to be clarified by the user so the algorithm can learn from. So the detection feedback can be thought of as detection confidence, which means how confident the algorithm at this moment think about the sample in the training image. And the user can look at the low confidence, which is identified in blue, and clarify whether it's a cell or non-cells. One might ask a chicken and egg problem here in our framework. Which comes first? Is good detection result come first, or good training sample come first? It's a very challenging problem to ask in many disciplines. But fortunately, in our case, with the size distribution and knowledge from previous cell type, initially we already give good enough detection confidence so that the user can focus on selecting a good training sample for a system to learn. And as you see, this kind of go round and round in iterations, and I will show you later on that with only 10 iterations, the algorithm will be able to convert to a good model and be able to detect cells. So for the experiment, we like to ask, is GATLab an accurate cell detection with only 10% of the training effort? First of all, we collecting our data, and this data actually come from our collaborator was actually interested in study. So we have 5 different cell types, and we're collecting 50 images of cells that look very different, and ask our collaborator to label them manually. The reason to do this is so that we know exactly where is the true location of cell. So when we compare to our method, we know the performance of the automatic method. So the collaborator is labeled over 2,600 cells. It wasn't fun for him, but he's glad that he'll help us. To compare our method, again, a series of algorithms that being used to detect cells. So we start with a baseline called adaptive boosting, proposed in 2000. The second method, task triad boost, addressing the second problem of first timer. So it learned from previous cell type. This method proposed in 2010. Our own research group come up with a method that addressing both the first and second problem. So we obtained the cell site distribution to address the first problem as well. This had been done in 2011. And until now, our new method, GATLab, had been proposed to address all three of these challenges. So we added select the most important samples. So let's see how these methods perform in respective to each other. So to perform the experiment, we're going to have a large number of training samples. We're going to be able to select only from 1 to 10 training sample out of I think it was 150 training sample. So we see which method be able to select these samples. And here are the results. Let me explain the exit. The x-axis is the number of training sample, and the y-axis is the accuracy. Basically from 0 to 1, and higher means better. Our proposed method, GATLab in green, had outperformed all other methods with training sample from 0 to 10. But if you look at how with only one training samples, with the help of side distributions and learn from previous cell type, our method and global triad boost already perform very decent, and that gives us a good leverage so that we can select the most important training sample. Then we can quickly reach to the convergence with only 8 training samples, and our accuracy is among the upper 80% in accuracy. All other methods, however, perform not as good, but we believe that it's just because our method be able to select training sample a lot more sufficiently than our other method. So we construct a second experiment to see how long it would take or how many training sample it would take other method to achieve a similar performance to our methods. In such way, we can know that how sufficient our GATLab will be able to learn. So we conduct a similar performance, but this time we let the method run until it converge, and we find out that about 100 training sample would be needed for other method to achieve the similar performance at GATLab. So let me show you here. It's a converger. So let me show you some images that have the visual effect of the cell detection method. To start with the natural killer T cells. With AdaBoost, you can see with 10 training sample was inaccurate. Our method is performed better. Similar result in the HT29 colon cancer cell line. AdaBoost with 10 training sample leave out a lot of cell undetected. Our method has fixed this problem. So an application that we use is into cell tracking. We're gonna track these cells under the microscope and see its performance behavior. And this actually, I focused on cell tracking last year and present at the graduate research fair about it. If you hear last year, you actually heard about it. So in conclusion, we have an accurate cell detection algorithm called GATLab, and we only require 10% of the training effort. Our research helped biology to study various cell types that can be benefit to cell analysis process and further help in cancer study. Our method be fortunately enough to result in several publications including one IEEE transaction journal and a recent submission to a top-tier computer vision conference, CVPR. And of course, I didn't do it all by my own, so I'd like to acknowledge my mentor and collaborators from UNC Charlotte, Carolina's Medical Center, and funding from the National Science Foundations. And finally, thank you for listening.